Good afternoon, and welcome to today's webinar, Interpreting Interim OS Overall Survival for Decision-Making. Overall survival remains one of the most clinically meaningful endpoints in oncology, but in many trials, earlier efficacy endpoints such as response rate or progression-free survival become available for review before OS data are mature. At that point, interim OS may still be considered to help assess potential harm and contextualize the overall benefit risk profile of a product. This creates, though, a difficult tension at times. How do we avoid overlooking a meaningful signal of harm while also avoiding overinterpreting immature and uncertain OS data? This work that will be presented today builds on discussions initiated through our annual meeting and subsequent white paper, which brought together a multidisciplinary working group to consider how interim OS could be more consistently planned for and interpreted in oncology trials. That effort identified the value of a more structured, simulation-informed approach and helped shape the work we are discussing today. We're especially grateful to the MMS team and our working group participants, FDA colleagues, and the many collaborators who contributed their expertise to advancing this project. Today, we'll hear an overview of the simulation framework and key findings, followed by a panel discussion on practical implications for trial design, analysis, and regulatory decision-making. I'm pleased to turn it over to Névine Zariffa of NMD Group, whose expertise and leadership have been central to translating this project from a conceptual framework into simulation work we are discussing today. She'll provide an overview of the framework and key findings. Névine, over to you.
Thank you, Mark. It's a pleasure to be here with all of you today to talk about the project overview and tee things up for the panel discussion, which I think you're going to find absolutely illuminating. So why are we here? We're here because patients are living longer with cancer, and that's due to advances in treatment therapies. Paradoxically, that creates a challenge for us for the next wave of therapeutic options. If we were to wait for fully mature survival data within those trials, it would take too long for patients. So what are our options? Well, luckily, we have good intermediate endpoints such as progression-free survival, disease-free survival, where we can make regulatory decisions around those endpoints. And at the time that we get the package for those intermediate endpoints, we would have a limited amount of interim overall survival that would be used in that regulatory decision making. So that sounds great, but what are the challenges? Well, the challenges is that it's early OS data, it's interim, we don't have the full follow-up, and we have few events. So statistically, this means that the estimates are unstable, and they're, of course, extremely sensitive to the timing decision on when we perform the interim, especially when we're talking about non-proportional hazards, where the early part of that Kaplan-Meier curve may not be what we see later. So now we need a good way to set ourselves up for the analysis and interpretation of interim OS. So luckily, we have a draft guidance from the agency. And the first and most important component is it says at the time of an interim analysis, where we're really looking at an intermediate endpoint and we have limited OS, that OS data is seen primarily as a safety endpoint. Lots of considerations laid out here. We're going to step through them in terms of the work we did and how we adapted our framework to address these elements of the guidance. And then they call out the value and the usefulness of simulation. And that, in fact, is the same conclusion that we had gotten to in our white paper. So Friends of Cancer Research, convening a number of sponsors, the FDA, academics, and importantly, the team at MMS that conducted the simulations for us. And so when you start doing work of this nature, what you're really looking for is a methodology that can be applied generally no matter the context that you're operating in. I'm going to call this a scaffold, others will call it a framework, others will say methodology, but here are the elements and the way we approach the work that would probably be useful to many others to adopt. So first ask yourself at the design stage a series of questions. Primarily, what do you expect to see in terms of survival for the control arm for your agent? And then because our hopes and desires don't always materialize, what are the plausible alternatives that you also need to catalog? Then look at the options that you have for decision criteria for identifying harm at the final once you have fully mature OS. And we do this because we need a stake in the ground to compare ourselves to. Once you have all of that done, you can use the simulation to evaluate operating characteristics at the final for that main scenario, the alternative plausible scenarios, and you could see if those decision criteria you identified perform well. And let's say they do perform well. Well, now we're finally in the zone of the question we asked ourselves is how would we judge the interim? And what are our tools and options for those decision criteria? And do they concord with the final? That's kind of the scaffold. So let's look at it step by step. First, in our work, we had to identify a number of these main scenarios for the final OS, as well as those plausible alternatives. We did that. We chose some. You can choose others, but I'll speak about these primarily today. First, you could have proportional or non-proportional hazard. The proportional hazard's a little more straightforward. Usually, we're going to think we have a main scenario where we expect to do really good things for patients, so a profound hazard ratio. Maybe the plausible alternatives, or it's a little less efficacious, or perhaps it's neutral, and the value is in the progression-free endpoint. And then for completeness, because we have to know what would happen with all the decision criteria, if we were surprised and there was harm, we include the harm scenarios. For proportional hazard, you're going to be looking at things where maybe it starts off strong and then the benefit on OS fades over time. Perhaps you're allowing crossover to the active agent. Or it's delayed. It takes some time biologically for the benefit to manifest. So in the early part of the curve, when you're going to be doing your interim, it's more neutral or dampened, and then it's later on that it would manifest. And we could have an early harm in a subgroup that's quite profound, and then that attenuates in the rest of the population. So those are the scenarios. After the scenarios, we talked about having decision rules. And so if we zoom into the top left of the table in the middle of the slide, we're looking at the final OS analysis, that thing we're going to test ourselves against. We chose three options. The first two are kind of deterministic. They work on the point estimate. First, we are totally uninterested in anything that suggests a hazard ratio above neutrality. Or we allow ourselves a little bit of leeway up to a 30% relative risk increase. And then last, maybe we remember that all these things are estimates that come with precision considerations. We look at the upper confidence limit, in this case 1.3, and we used a 95% confidence interval. So those are the three decision criterias that we used for the final OS. And then the flip side is that the interim, we were interested in the toggle between upper confidence limit thresholds ranging from very, very tight, 1.1, all the way up to 1.8, and you can even push that further. And the relationship between those threshold choices and the amount of information we would have at the interim for overall survival, very little, 10% of the overall data coming in versus 50%. So now that we have all of this laid out, what's the process by which we evaluate the operating characteristics? So first we ask ourselves, for the number of events, for the thresholds, for the decision criteria, actually, even at the final with all the data in hand, how often would the trial decision criteria line up with the scenario that we've picked so we know what it is so we can judge. If we have a good rule there, we go to that next step, which is, does the interim agree? And if it doesn't agree, is it because it suggested harm when it shouldn't have? Or is it because it failed to see it and it was manifest later on in the final? So let's go to the final OS and those decision criteria and how they perform. So what you have here is one of many different tables that we've created through simulation work at MMS. This particular one says we're not in the indolent setting, we're not in the advanced metastatic, we're in intermediary, where at the end of the day, we will have 150 events. We set a control rate for those OS events of 0.2. I focused here for simplicity on the proportional hazards down the columns from left to right, benefit all the way to harm. And what we're looking for is white. We want those cells to be white. We don't want to see black per se or gray. And we have the point estimate of 1.0 on the top row, then 1.3, then the upper confidence limit at the bottom of 1.3. And using this plus all the rest of the tables for other event rates, for more events and the indolent setting and the non-proportional, the overall conclusion that we reached is that that middle row with the point estimate of 1.3 just doesn't do well overall, this variety of scenarios. So we're looking at the first row or the bottom row, and we chose to pull through the upper confidence limit just on statistical principles that we wanted to include the precision of the estimate in our decision criteria. So now let's apply all of that back to the OS at interim. We have our 150 for final. We have our event rate of 0.2, same as the previous slide I showed you. And what I have now is a heat map. And this heat map, we'd like to be in the richer, deeper blues. And we don't want any red. And the columns will tell you I have very little information, 10% on the right-hand side. All the way through, I've waited to conduct my interim to the halfway mark of the event accumulation for OS. And the rows now starting at the bottom is very, very tight. I want to be very cautious when it comes to declaring that there's no harm at the interim, all the way to I recognize that this is interim and I have to widen my threshold to say 1.8. Takeaways from this, I think you will recognize immediately. 10% is just not enough information. Even if the actual scenario is a strong OS benefit at the end of the day, at 10%, we do not make reliable decisions. So I'm looking maybe for the column at 30, certainly 40 and 50. And now I have a trade-off. If I wait longer, I can have a tighter threshold at my interim. But the reality is we're going to want to go as early as is possible, and we can get reliable interpretation. So that's in the 30%. And really, we're looking at thresholds that have to be a little higher. So the 1.7, the 1.8. Now, remember we said that we wouldn't set out a main scenario where we have this benefit, but what if we're totally surprised and the data tells us there's profound harm? Well, the good news is that no matter what, we're going to pick it up here. So that's not really a consideration in terms of where we might pick the timing or the threshold. But what is a consideration is when you look at those non-proportional hazards, remember I said they're a mix of what happens early and late. So now the amount of information fraction is linked to the duration of follow-up. And what you have here on the left-hand side is it starts off strong, very much like that proportional hazard that I showed you in the very first example. And so the blues and the reds are quite comparable, and you come to the same conclusion. But what often happens is I might think, look, I'm going to start off strong, I'm allowing for crossover, it's going to dampen. But what's really happening is that you're going to have to wait longer So you have the right-hand side where it's a delayed benefit. And there it's very clear it's a 40% information fraction or 50, and you're looking all the way up to the 1.8 as the threshold. So that said, let's take a step back and let's start to pull it all together for the panel to come. So first, it's really interesting to know and to be clear that even with all the data in hand, across the different scenarios and the plausible truth, the definitions, the decision criteria don't perform equally well. So that's something to really have at the back of your mind. We know that at the interim, we're conducting a trade-off. We're saying, how long can I wait? And then how certain do I want to be? And how rigorous do I want to be in terms of setting that threshold? And last, it always depends on the context that you're in. The survival pattern itself, the number of events, your ability to have full coverage in terms of the follow-up period or the crossover, and that pesky non-proportional hazard that we spoke about. So my last slide before I turn it over to the panel conversation with Briggs and the rest of the group is, in practice, you got to plan. You've got to plan, and you've got to plan in a very rigorous fashion with respect to all of the elements that we've talked about. Once you've pressure tested those elements, you're going to pre-specify and you're going to justify. Sometimes it will be on statistical terms that the threshold set any lower just isn't reliable. Sometimes it will be clinical. Sometimes it will be that, oh, I'm going to abandon this setting of a threshold in the statistical sense of the word, and I'm going to revert to using OS as part of totality of evidence, where it's interpreted alongside the efficacy of that intermediate endpoint, alongside the other safety in the context of follow-up crossover and the clinical context. So on to the panel discussion. Thank you all.
Névine, thanks so much for your presentation. That was really, really well done. I'm Briggs Morrison. I'm going to be the host of the discussion here today. I'm a medical oncologist, spent many years both in large pharma and small biotech. I want to thank the Friends of Cancer Research for coordinating this effort, everyone who participated in the work, and let me introduce our panelists. So first we have Andrew Mills from MMS Holdings, a statistician who did the bulk of the simulation work that you'll see in the program and that Névine briefly summarized. Kiran Patel from J&J, a practitioner of both designing trials like these and interpreting the data. Lisa Rodriguez from GSK, interesting background in that she's been both at the FDA and in pharma, and if I understand correctly, was actually the primary author of the FDA's overall survival guidance document. And Xu Wang, who is currently a statistical reviewer in CEDAR. So that's our panel, and let's jump right into the discussion. So let me start off with the project was motivated by the recognition that interim overall survival interpretation often lacks a consistent framework. And so maybe for the panelists, from your perspective, why is it important to approach this challenge collaboratively? And where do you think the alignment across the stakeholders is most needed? And maybe we'll just start, Karen, with you.
It's great. Thank you, Briggs. And thank you for the opportunity to provide the perspective of, this very interesting but also very challenging topic in drug development. I would like to just level set, to all of us that, overall survival in any aspect, remains a gold standard endpoint, for patients, for regulators, for peers, and also for the industry, given, the impact, we need to continue to make for our patients, who are suffering from cancer. But from the development perspective, the challenge is really particularly critical due to several reasons, especially when it comes to interim overall survival. And it starts with most of the time having insufficient follow-up, long post-progression survival, multiple subsequent therapies, that these patients go through, and also potential treatment crossover, that happens, through almost all the time. And in this setting, in this kind of setting, the early interim OS read can be confounded. I think we all know that, it could also be significantly underpowered. And sometimes it's very difficult and misleading potentially to draw any actionable conclusion based upon this. I would also highlight the fact that in advanced cancer, interim survival, may be in some ways achievable through significant follow-up over time and waiting for the survival data to mature. I think it becomes even significantly challenging as you go into early cancer, where the risk benefit is very different for patients, especially in breast cancer or prostate cancer, even breast cancer, where it would take 10 to 15 years, for these patients to, to have any meaningful difference. And in really, this kind of interim survival often lacks, this consistent interpretative framework that allows us to really, meaningfully, interpret the data. So I think to your point, Briggs, the collaborative approach is critical to ensure that the interim analysis are interpreted within the appropriate clinical context and also aligned with the totality of evidence, including alternative endpoints that actually allows us to, appropriately measure at that time. And the alignment is critical and is needed across all functions, clinicians, statisticians, regulators, and also sponsors on the expectations on interim data maturity. How do you handle the confounding factors and the potential role of complementary endpoints in decision making to ensure that there's a consistency in interpreting the data?
Super helpful. Thanks so much, Karen. Lisa, I'm curious your view of that question. Why is collaborative approach so important?
Great, thank you, and I'm happy to be here. I agree with many of the comments. It has been challenging to assess OS when it's not the primary endpoint. So often it's immature and there's high uncertainty at interim. And planning for in advance for assessment of harm or the no detriment was one of the key points of that new OS guidance that came out last year. But how to define harm and how to talk about it in a consistent way has not been clear. So understanding the clinical and regulatory context, as mentioned, is very important. And the definition of harm and the expected timing for the interim analysis is, of course, going to be informed by the disease, the intended population, line of therapy, safety profile, and other issues. So collaboration amongst the statisticians, amongst the clinicians, talking to the regulatory colleagues, and also internally understanding the other stakeholders in development for a sponsor is necessary early when designing the study, defining harm, and thinking about when you can assess and what is feasible. What's a feasible interim, time point where you can potentially make decisions and inform development and have discussions with stakeholders and with regulators. So, of course, I think that's really a multidisciplinary effort. Having that discussion is important to plan the study as well as to interpret the data at interim. And really being able to make informed decisions about the presence of harm or deciding if the study should be stopped should be a multidisciplinary discussion.
Mhm.
Thank
And
you.
Shu, maybe your perspective from your work reviewing applications and study designs at the FDA.
Yeah, sure. Thank you for the question. And so from regulatory perspectives, OS is an important endpoint to inform both efficacy and safety. And the requirements for interim OS looks may be different by disease settings and drug classes. So from our perspective, we feel that it's really important to communicate and reach alignment between the agency and the sponsors. Thank you.
Super. So maybe Andrew, we'll start with you on this next question. So what do you think are the biggest challenges or points of uncertainty around an interim OS interpretation from your perspective?
Thanks, Briggs. And as you mentioned, I was involved quite heavily with the team in MMS getting the simulations together. So I'm going to try and step back and keep it quite high level. And it really is a multifunctional problem. So if we think about at least one of the bigger challenges, it's the timing, the timing of the interim. And it's difficult because we've got multiple different factors pulling this timing in different directions. We've got elements that will pull it earlier, so we want to avoid any undue exposure of participants to a truly harmful treatment, but also pulling it later. We want to make sure we've got enough information, data is mature enough that the estimates and the confidence intervals around them that we get are precise and we can make sufficient conclusions, especially when the treatment's truly safe. And there's an extra element of it's just much more operationally simplified if we perform the first interim OS at the time of a primary readout for a surrogate endpoint such as PFS. But if we haven't evaluated what this means in terms of information fraction or even just the conclusions that we can get in OS, we're really setting ourselves up for a bit of uncertainty. So I'm really thinking this drives us to an ultimate question of how early is too early, and in particular in the context of assessing the hazard ratio of overall survival as a measure of safety. Because we do have other measures of safety. We've got adverse events, we've got safety labs, but understanding at what point do we tip from an imprecise or an unpredictable to a meaningful and useful measure of safety using the hazard ratio from overall survival. Really
And
just understanding that
in Navene's
immaturity.
in Navene's presentation, she talked a little bit about different scenarios about what the information fraction is. From your work done and all the modeling, is there sort of a minimum threshold that people should be thinking about that it's probably just not informative below a certain information fraction?
Yeah, it's a great way to think about it. It will depend partly on the disease area, the mechanism of action of the drug, and the anticipated and plausible alternative effects. But what we're really saying, anything lower than around 30 OS events, you're setting yourself up for failure. There will be extremes, so there will be settings where the effect size, either on the negative or the positive side, will be substantial enough to determine that. But that's really kind of what we're seeing across the board.
And Navene, what would you say are the biggest challenges or points of uncertainty?
I like everything I heard from Andrew, and I'm glad that you asked that question about like, is there a no-fly zone minimum below this? It's not the statistics that are going to help you. It's that good old-fashioned kind of clinical interpretation. But to answer your question, I believe that it's this kind of odd thing, like we forget it's interim, we forget that it's limited, and we look at it through deterministic lenses. We think this is it, this is what we've got. We've got to draw definitive conclusions. And the work we've done tells us, no, it's not that simple, and you could easily mislead yourself. So the tools that we set forward and that I gave a snapshot of really help us understand what is the level of confidence we should have in our understanding of what the interim OS can reliably tell us. And we've just heard from Andrew very elegantly, it's a trade-off. Too early, not so great. Too late, not so great. Being overly stringent or overly permissive. And that's the tension and the trade-off in the regulatory review process that we're going to want to consider for approvability. And it's interesting that the statisticians are all saying this, but there are times when it's not going to be quantitative or statistical. And that's the big thing that we should remember. I'm not going to go back through everything that Andrew said, but let's make sure that we remember that. And then the last thing, operationally, it's a choice that we make to anchor to that intermediate endpoint and its maturity and doing an analysis at that point. That could have consequences on our ability to have reliable interim OS. So if that's critical to the decision making, you could consider other choices for that timing of the interim.
Great. So I think, as Andrew pointed out, in the past, many people have really just used a quick look at OS at the time that the surrogate endpoint reads out, and that may or may not be sufficient. So maybe Lisa, you can start us off. How do you think the field can do a better job of sort of prospectively planning how and when to interpret interim analyses?
Thanks. So again, I'll mention the guidance, which I will say was very much a collaborative effort between the clinical and statistical colleagues. And in the guidance, of course, it mentions to plan, but how to do that, I think this Friends of Cancer work is very helpful. As Névine went through, there's different scenarios. Understanding the uncertainty associated with those different scenarios, thinking about when potentially to do that interim, I think can be very helpful from this work that was done. And the sponsor, I think, can also really think perhaps about a decision framework when they're planning the study. How are they going to make decisions? When might they make decisions? What risks are acceptable to them for their particular study or development plan, their product? So that's going to, of course, define the scenarios you're looking at and what decisions need to be made. They'll need to think about what's feasible for them, feasible sample sizes, and timing. I think sponsors will need to think about incorporating their risks, risk to running the study, risk to, potentially making a wrong decision one way or the other, and really thinking about the level of uncertainty that's acceptable. And they'll need to think, again, amongst the multidisciplinary colleagues, how to define harm, and what's relevant to their program and, their known safety and their planned approval pathway. So I think all those things will come into play, and perhaps that could be thought of in a framework in advance.
I am just curious, I don't want to put you on the spot since you are at GSK, but do you have internally at GSK that type of planning process for your studies? And do you have a framework that you guys use internally?
Um, I think there's always a discussion about the risks and the different approaches and the timing. Um, I would say it's probably a work in progress in terms of do we have a specific framework and a specific way to think about OS and interims. It's certainly in discussion, and I think this work is going to be very informative for that.
And Kieran, maybe just ask you that same question. Do you guys have within J&J or within your group a framework of how you use this in the planning process?
Yeah, Briggs, that's a great question. And I think as we take different approaches, for different patient population, we are soliciting, publications and presentations to come up with the framework. The Friends of Cancer Research work that is just completed now, will be another tools to apply. But to your earlier point, I think to Lisa's point as well, it hasn't been routinely done for a lot of different reasons, especially when we don't expect that survival is going to be critical. The study is not powered for that purpose, and you don't have sufficient events that we normally do not put statistical assumptions surrounding it. I think it's becoming more critical now to include that as part of the statistical assumptions upfront, proactively into the protocol, have alignment with the FDA to that framework that we would want to consider for the study and get that alignment upfront. So I would say work in progress, just like Lisa, but definitely taking into consideration different methodologies, including the one that Friends of Cancer Research is coming up with now, so that we can proactively, implement those framework in the protocol.
And Névine, what about your thoughts on how the field can do a better job of prospectively planning?
It's I think what we're hearing is there's intention, but there's also variability in that planning. So I think Lisa used the word work in progress. My own personal experience and my prior companies was that it kind of depended on who was at the governance group asking you whichever question that it might have been. The discipline and the comprehensive approach that we've put forward here, I think if we can apply that across the board, there's nothing magical about it. There's nothing prescriptive about it. But if we say, okay, so let's start off with what's my best hope for this drug? What's my main scenario? Why is that? Because I've looked at all the context data that I have at that point, natural history, control arm performance, other features, other agents in the class, what have you. And I lay down, physically write down, these are the plausible alternatives. Just that. just like that, would be quite relevant. Lisa pointed out a number of other components that have to do with the decision, the threshold, the probability levels, the timing, the expected number of events. All of that could be laid out and studied. And if everybody did that and it was structured that way, my God, we would learn as a community so much faster. And I think this would pay off in multiple different ways, and we can talk about that a little later. So a bit of discipline, maybe adopting like the framework that we've put together. You can add to it, but let's say at a minimum you would commit to doing that. And then sharing so we can all learn from each other faster.
Shu, I'm curious from your position at the FDA, have you seen since the publication of the guidance document, sponsors coming to have these conversations with you prospectively?
Yes, I think for sure. We have seen studies and protocols, including OS harm looks and also sponsors talking to us to discuss how to appropriately better design the OS interim looks and identifying for harms. And I think, as we mentioned, many panelists, we all agree it's a multidisciplinary work. And also, as mentioned earlier, I feel from regulatory perspective, there are many considerations. So we don't really have a one size fits all or one method fits all on our side. So yeah, we will review all the aspects, involve all the disciplines, and to engage with the sponsors and to better discuss how to appropriately design OS harm looks. Thanks.
And what types of information, Shu, do you find to be most helpful for the sponsors to bring when they're having those conversations with you?
Yeah, sure. I think that's a great question. So I feel before I go into the details, I just want to mention that. So all the studies are different in terms of many aspects, like study design, the cancer types, the target population, the study drug classes, and so on. So and just want to cover some information for general considerations, but each study definitely needs its own specific considerations regarding the OS monitoring. So generally, we feel one aspect that would be helpful is the detailed information provided by the sponsor. And by detailed OS information, that includes the expected number of OS events at various time points, including at PFS readout, at OS monitoring time point, and as well as at OS final analysis, because such information could really help the agency to better understand the maturity level of OS data throughout the conduct of the study. And another piece of information that's also related is the operating characteristics of the proposed OS monitoring rule. So the operating characteristics are usually obtained through simulations, and those information is also very helpful for the agency to better understand and evaluate the appropriateness of the proposed OS monitoring rule. And there are some other information that could also be helpful depending on studies. For example, the anticipated subsequent therapy and its impact on OS data, and whether the crossover is allowed. If it is allowed, what is its impact on OS data and some other aspects. So those are some general information we feel it would be helpful in the discussion. But again, just want to mention those are for very general considerations in our past experiences. And for each individual study, the sponsor should definitely discuss with the corresponding review divisions regarding what information would be the best for their specific study in the discussion. Thank you.
And Kieran, maybe same question for you. What information have you found to be helpful when you guys go to have these discussions with FDA about interim analyses of overall survival?
Yeah, I do think that we usually take a very simple approach of quote unquote designing a statistically powered survival, which most of the time at the time of interim look is almost impossible, to power it. And so it in some ways discourages sponsors to even put any kind of statistical assumptions, at that time, which again, I think is counterproductive, from the perspective of understanding, the potential risk part because the benefit may not be clearly interpretable at that time. And so, most of the time, I think, to that point, we try to bring in, more granularity or more specificity around what the expected percentage of events would be, and maybe put on some level of, potential no harm, type, threshold, to be able to at least rule out the harm rather than potential, looking for the benefit. And working in partnership with the agency, we find that quite helpful, in some cases, right? Where the events are very small numbers and hard to really make any conclusion, positive or not positive at that stage.
Yeah, super. Thank you so much. So, of course, one of the goals of the project was to provide this more structured framework for evaluating this. And maybe, Lisa, you could just share from your perspective, where do you think a framework like this is most useful in practice?
Great. So I think that the work that Friends of Cancer did, evaluating the different ways to think about harm, thinking about point estimates or confidence intervals and different thresholds, are really very applicable to oncology studies to help think about harm in meaningful and applicable ways. Um, I think sponsors will need to think, as we said before, planning what definition and threshold may be most meaningful for this particular study that they're planning. So perhaps for a phase 3 pivotal study that will be used for regulatory decision making, there could be a need to address a safety profile, and use more stringent thresholds for harm. It could be that there's already a known positive safety threshold or safety profile, and you really just need to show no detriment and have a much less stringent threshold. So it's really going to depend on where you are in terms of where this study fits in for regulatory decision making, and what's known about the product, and again um, your own thresholds for risk. It may be the case, of course, that the regulatory agency has some thoughts about what that threshold might need to be, and what the definition would be. So in either case, I think, again, the definition of harm, the timing of harm, um, needs to be determined in advance and informed by the clinical and regulatory context. And you can really have, I think, some good discussions both internally and with regulators around some of those scenarios and choosing the threshold, and perhaps looking at some of those tables, like Névine showed. Thank
Super.
you.
Andrew, your thoughts of where this framework could be most useful in practice?
Yeah, I was at a conference this last month for statisticians in the pharmaceutical industry, and I was in a really interesting talk about independent data monitoring committees, and it made me think we have this third party, and especially when we're talking about hazard ratios, if you want to maintain trial integrity and blinding, we're going to have an independent group evaluating that comparative data. And that's already a pretty tricky space to navigate because sponsors are essentially handing over the ultimate well, not the responsibility, but the ability to determine whether a trial continues or stops to a different group. Um, and whenever we add on this layer of uncertainty around what is harm, and we leave it to the intuition of a different group, we get misalignment. And now we have misalignment potentials between the sponsor and their IDMC, the sponsor and the regulator, and then the regulator and the IDMC. So it's just a multiplicative problem. But if we can lay out quantitatively what this looks like through a framework such as this, we allow sponsors to write IDMC charters that have decision rules specified within them. Obviously, there's going to be an element of expertise that gets added on with the series of information that supplements beyond just a hazard ratio or the confidence limits around that. So what it does, it gives sponsors this ability to write this information and pre-specify it, most importantly. But just to add one layer to that, it gives smaller groups. So if we extend slightly beyond mid to large pharma, small biotechnology companies are empowered to create and define these what can be quite scary concepts if you don't have a group of statisticians more easily, or at least using, independent bodies. And it gives them some confidence in, well, what does this mean as somebody who doesn't understand statistics in a slightly more grounded or clinically relatable fashion. So I think that just the widely availability of that information to different groups like IDMCs and then smaller companies is going to be a big help.
Excellent. Thank you. Névine, what do you think? Where was this framework most useful?
Well, I think number one, let's just say we're going to have the framework. Let's just do the framework. Let's have all the elements of the framework outlined. Let's get that commitment that every cell, every component of the framework is going to be considered. As Shu mentioned, we don't want to be prescriptive around the decisions or the arguments or the assumptions the sponsors will make, but we don't let them get away without considering every aspect of the framework. So that's already one step forward. And I think if we do that, what we're going to get are regulators are going to make more consistent decisions. We're not going to be scratching our heads going, well, why did this sponsor get this outcome and this other one with roughly the same data get some other outcome? That's not good for patients. And so I think the regulatory consistency across the many different teams that work at the agencies around the world, that's helpful. And here's the last thing. We're not doing this to have panel discussions and papers and stuff. We're doing it for patients. So if the patients could see the elements of the framework, kind of translated appropriately, and for any trial of any drug that they were considering taking, they could say, okay, here's what they thought, here's what they were hoping, this is what they also considered, here's what the data was, here's what the outcome was. They would probably make more clearly informed decisions for themselves. So I would say it will help with consistency of decision making within the agency, which we don't always have. And then it will help in terms of informing patient choices.
So, Lisa, maybe next question for you. Again, given your unique background of being sort of both on the regulatory side and now on the at GSK, the question is sort of how do both sponsors and regulators balance that risk of overinterpreting immature data on the one hand, but on the other hand, missing a potentially meaningful signal of harm. And you've looked at it from both sides. I'm curious how you think about that.
Yeah, I mean, I think that the work that was done here is very helpful, but I do think additional work from everyone will be needed to look at these very specific scenarios and really think about where are we in the type of decision that needs to be made based on the known safety and the drug and the regulatory decision and everything. I think more will need to be done there, perhaps specifically by the sponsor. It would be nice if as was mentioned, people share knowledge and inform everyone about some of these different scenarios. I think if sponsors and regulators, though, are not willing to accept the risks and uncertainty, then a lot more work will need to be done. And those will need to be internal discussions as well as you're planning the study and have those multidisciplinary discussions and talk about uncertainty and talk about risks and understand that if you do have immature data, you will have a risk of making a wrong decision. So which way is less risky, I guess, for you in terms of the decision you're going to make and what's feasible in your drug development timeline and what decisions you're going to make that are both going to inform this study and potentially inform other studies for the sponsor. So I do think, thinking about this in advance, talking about decisions, the timing for multiple decisions that need to be made, what will inform each other and what levels of risk are acceptable. And then speaking early with the regulators when planning and making sure that there's some agreement about, definition of harm, level of uncertainty, and what sort of thresholds will be used, I think will be very helpful to have those discussions early.
And Shu, I imagine this comes up with you and your colleagues on a not infrequent basis, the same question of, are we over-interpreting interim data, but yet we have to balance this risk of harm. Maybe any thoughts you have on how to think about that?
Yeah, sure. Yeah, this is really a good question. And I will just start with two examples representing different scenarios. So the first example is the Bellini study in relapse refractory multiple myeloma. At the study interim, it reported an OS hazard ratio of 2.03 with a confidence interval from 1.04 to 3.94. And the number of deaths in two arms were 41 versus 11. So the results of OS showing a clear OS detriment. And however, the chance of identifying the early harm signal and potentially terminating the trial early was missed. So this example really highlights the need of early monitoring for harm in the design stage. And another example I want to mention is the Checkmate 6202 study, also in relapse refractory multiple myeloma. So prior to going to the detail of this study, I want to mention the background is prior to identifying any signal of this checkmate study, the agency actually required another two pembrol study. One was Keynote 183, the other was Keynote 185. The agency required those two Keynote studies to be discontinued due to the safety concerns and lack of evidence for efficacy in 2017. And then the agency also put the Checkmate 602 study we mentioned earlier on a partial hold, also in late 2017 due to the safety concern observed in the pembrol studies and also the unfavorable risk-benefit ratios. And later, the agency lifted the partial hold in 2018 for Checkmate study. But then the sponsor, after the sponsor amended the study protocol to address the toxicity. But the sponsor conducted a futility analysis and eventually decided to discontinue enrollment from their end. So in this example, due to the concerns identified of another drug in the same class, the early signals was identified and the Checkmate study was terminated early. So I think to balance the risk of interpreting immature OS data against meeting a potential OS harm signal, many aspects should be really considered. So one is IDMC's regular monitoring, considering closer monitoring if deemed appropriate, because the IDMC will review not only survival data, but also many other safety data and safety endpoints. Another thing I want to mention is to be aware of the specific safety concern of a drug class, like the above mentioned Checkmate example. And last but not least, as I think almost every panelist mentioned, it's really a multidisciplinary effort, and we should really consider totality of data when we interpret OS information. Thank you.
Super helpful. Thank you so much. So maybe the last question for everybody on the panel as we look ahead, where do you think the biggest opportunities remain to improve the design, interpretation, and communication of interim overall survival analyses? And maybe we'll start with you, Kieran.
Yeah, Briggs, I really think that the work Friends of Cancer Research has done is really a great start to really put some framework around this. But I also, as a drug development optimist, we are moving in the direction where we are making a huge impact in patients' lives. Survival remains the key, part of that, that achievement. As we move into the earlier disease, where we can make even a bigger impact for patients. I think this is going to become even more critical on how we define, how we really look at safety, and maybe in that context, safety will be survival will be a safety endpoint. And so there has to be more work to be done here. I think it's a great way to start, but also I wouldn't consider this as the end. I think there are a lot of other methodologies that can be implemented here to really help the industry, but also the regulators, FDA, to feel comfortable with those framework and ultimately help to really get the most impactful drugs to our patients as fast as we can.
I like that drug development optimist. I think we're all drug development optimists on this call. Andrew, your view, what do you think the biggest opportunities are going forward?
I see it using this framework or something similar as a targeted simulation exercise to look at when is it that interims applied the most weight, or where can we apply the most weight to different interims? So where is it we can gain the most information and make the best decisions? And really as a maybe thinking more about the statistical aspects as a group, just being more open and improving and spreading the capability of these techniques, and then making available things like this framework and the associated programs that go into the simulation. With the ultimate aim, as Névine had already mentioned, we're trying to get patients safe and efficacious treatments as soon as possible. And if we can do something to assist others in getting there, it just makes sense.
And Lisa, what do you think the biggest opportunities are that remain to improve the design, interpretation, or communication of OS analyses?
I think something that's come up is really like a decision framework is probably needed, something that you have a good interdisciplinary discussion, it internally really think about your acceptable levels of risk and what decisions are going to be made, as well as the context, so the regulatory context, the development context, and what other data you have besides OS to assess safety. And I think putting all of that together, you're going to have a lot of different scenarios, and you're going to have something that's probably very particular to the sponsor's development program. So I do think sponsors will need to think about how they're going to put something like that into work for them. So.
And Névine, you kicked us off with the overview of the project. Maybe you can close us out with your final thoughts on where you think the opportunities are.
Thank you. And thanks everybody for a fantastic panel discussion. So here's my roll up of everything I've heard today from my colleagues is we're development optimists, and I'll add, we want to go fast and we want to go well. So we want to speed this whole thing up. We're able to come together as a community, lay down tracks, give general guidance. I think we got to move faster. We've got to turbocharge this thing. So I am all for some kind of systematic learning environment. And let's get the assumptions that we're making, the discussions that we're having with the agency out there in the open. Let's have Friends of Cancer Research, extremely good at mobilize us so that we can get and evolve to standards more quickly. Because it's not going to do us any good to say every single thing is a special case. No, there are going to be some things that we can take off the shelf, and this is what you're going to apply. If you want to deviate from it, that's your special case. So I'm all for going faster, doing good work well. I think sharing more openly against a good enough framework is going to be part of the solution set.
Yeah, super helpful. And I think Shu gave a couple of nice examples of sort of real life examples of cases that the agency has dealt with. And it may be worth, again, maybe Friends of Cancer Research can help facilitate this. Some of these more challenging interpretation, it may be worth having case studies of how sponsor thought about it, how the regulators thought about it, how patients thought about it, and sort of where we landed. So again, let me just thank the panelists. First, again, thank everybody who was involved in the project. Thank the panelists for your wonderful contributions, and I'll just turn things back to Mark to close out the meeting.
Thank you, Briggs, for moderating a great discussion. And thank you to Névine, Andrew, Kieran, Lisa, and Shu for a terrific panel conversation. Today's discussion reinforced that interim OS interpretation cannot rest on a single threshold or isolated analysis. It requires prospective planning, attention to the maturity and context of the data, and a clear understanding of the trade-offs involved in evaluating potential harm. We're developing a manuscript that will describe the project findings and recommendations in greater detail. We're also working on additional practical resources discussed today that can help facilitate prospective planning, communication, and structured consideration of interim OS and oncology trials. We invite you to follow the project's progress through the project page, where you can access the project overview document, today's webinar slides, and additional outputs as they become available. Please also feel free to share these resources with colleagues who may be interested in this work. Thank you again to our panelists, collaborators, and everyone who joined us today. We look forward to continuing the conversation and advancing this important work together.